Hello everyone, my name is Chen. Today I'm going to share a paper named Optogenetically Engineered Lactose Operon Repressor LACI Responsive to Light instead of IPTG. This paper was published on Nucleic Acid Research. So firstly, we should focus on the research backgrounds. The lactose operon is a classic gene regulatory system in E. coli, which can regulate the cellular metabolism. The components are divided into structural genes and regulatory elements. The LAC-Z can break down lactose into glucose and galactose. The LAC-Y can translocate lactose into the cell, and LAC-A has no function. And the promoter is the RNA polymerase binding site that initiates transcription. The operator are binding sites for LAC-I repressor protein that controls the switching of genes. And LAC-I, the regulatory genes, encode the LAC-I repressor protein, which can bind to operator to repress gene expression. So now we focus on the figure. In a case when lactose is present, lactose is converted to allolactose when it enters the cell. The heterolactose binds to the LAC-I repressor protein, resulting in a conformational change and detachment from the operator. The RNA polymerase is able to successfully bind to promoter, initiating transcription and expression of LAC-Z, LAC-Y, and LAC-A genes. However, in the absence of lactose, the LAC-I repressor binds to the operator, and RNA polymerase is unable to bind to the promoter, thus preventing the initiation of the transcription of the genes, and the gene expression is repressed. And this figure can be more graphically illustrate how the lactose operon in E. coli regulates the expression of the relevant enzymes based on the presence or absence of lactose, leading to efficient utilization of lactose. When in the absence of lactose, LAC-I inhibits protein binding manipulators, preventing RNA polymerase transcription, and gene expression is inhibited. But when in the presence of lactose includes the inactivation of repressor protein. RNA polymers initiate transcription, and genes are activated to the synthesize enzymes that metabolize lactose. The IPTG, which is commonly used artificial inducer of the LAC operon system, it has the following characteristic. First, the structure is similar to the lactose, which can mimic the induction effect of lactose. Second, ethanol is hydrolyzed by beta-galactosidase, which is encoded by LAC-Z, so it can exist stably for a long period of time in the cell and continue to induce the gene expression. So this figure can demonstrate the mechanism of the systematic regulation of gene expression under IPTG induction. In the absence of IPTG, the LAC-I repressor protein binds to the operator. The LAC-I repressor protein prevents RNA polymerase from binding to the promoter. This can result in the inability of the target protein to be transcribed and translated, and the gene expression is inhibited. The IPTG as lactose is able to bind to the LAC-I inhibitory protein. When IPTG is present, the LAC-I repressor protein is inactivated by binding to the IPTG, thus detaching from the LAC-O operator. the RNA polymerase can successfully bind to the promoter and initiate the transcription and expression of the target protein genes. However, IPTG has some significant drawbacks. First, its high synthesis cost can significantly increase the economy burden in large-scale production. Second, high concentration of IPTG may have an inhibitory effect on cell growth, and the accumulation of IPTG in cells may also lead to decreased growth rate or metabolic disorders. Finally, the induction of IPTG is irreversible. Once it is aided, the LAC-I inhibitory protein is inactivated and gene expression continues, lacking flexible dynamic regulations. So these problems limit the application of IPTG in industrial production and fine regulation. So the aim of this study was to develop a light-controlled LAC-I that utilize light signaling instead of IPTG for gene expression regulation. Most important in this article are these two optogenetics-based light-controlled gene expression systems. On the left is a light-induced system in which opto-LAC-I undergoes a conformational change under blue light irradiation, detaches from the manipulator, and the RNA polymerase can bind to the promoter to initiate the GOI expression. And light activates gene expression and darkness inhibits gene expression. On the right is a dark-induced system, where the opto-LAC-I detaches from the manipulator and RNA polymerase bind to the promoter to initiate GOI expression of the target gene in a dark environment. The difference is darkness activates the gene expression and light inhibits the gene expression. First, this is the structure of the LAC-I. The key structure domains are labeled in the diagram. The DNA binding domain is used to bind to the manipulator LAC-O and inhibit downstream gene expression. The oligomeric domain is used for protein-to-protein interactions and is located after the position 316. And loop 1, loop 2, loop 3 are the flexible loop region of LAC-I. And these regions are used as a sites for insertion of light-sensitive modules. The structure of LOV2 is shown here. The PS domain is the photoreceptor region, and the J-alpha helix is a key structure for light-sensitive signaling, and the CP-LOV2 is a modified LOV2, which is easy to fuse with LAC-I. Therefore, we can find the working mechanism of opto-LAC-I. Opto-LAC-I, the light activated, so in the light, LOV2 changes conformation and the DNA binding ability of LAC-I is inhibited, leading to GFP gene expression. And the opto-LAC-I dark type, when in the dark, LOV2 maintains its original conformation and LAC-I binds LAC-O, inhibiting GFP expression. So we can find there are two systems mentioned in this study. The first one is a working principle of opto-equalize light system. So when in the dark, opto-LAC-I is activated and firmly bound to LAC-O operon. This binding inhibits downstream GFP promoter expression. Exposure to blue light causes a conformational change in the photosensitive structure domain LOV2 in opto-LAC-I, which can activate the promoter and drives GFP gene expression. The promoter is activated and drives the expression of GFP gene, upon which the cell fluoresces green. So the first figure demonstrates the effect of lack of operon copy number on gene expression. Under blue light condition, the level of GFP expression was significantly higher than that under dark conditions, indicating that light can effectively release the repression of genes. With the increase of lack of operon copy number, GFP expression gradually decreased. It can be found that the higher the number of operons, the stronger the effect of genes repression. And the figure on the right shows that the effects of blue light intensity on gene expression. With the increase of the light intensity, the level of GFP expression increased significantly until it reached saturation. And in the figure D, we can find shows the effect of light intensity and times on gene expression. So under all blue light conditions, the GFP expression level increased rapidly with time and peaked at about 8 hours. When the light intensity was reduced, GFP expression decreased with the decreasing light intensity. And the graph E is the effect of the IPTG concentration on gene expression. So with the increase of the IPTG concentration, GFP expression increased significantly. At 0.1 millimole IPTG, GFP expression peaked rapidly. The induction effect trends saturate at future high concentration. Comparing to the opto-E. coli light system, the opto-E. coli dark system has the opposite response mechanism. Activates gene expression under dark condition, while inhibiting the expression of target gene GFP under blue light irradiation. Further, the researchers used the effect of different experimental conditions on gene expression. In graph B, the level of GFP expression was significantly increased with the increase of lack of copy number. Under the dark condition, it indicates that increasing the operon copy number enhances the inhibitory effect of opto-LAC-I on GFP. And the figure C shows the effect of testing different mutants of the opto-LAC-I protein on gene expression. Finally, the size specification K84 mutant was found to have a significant effect on the photocontrol performance of opto-LAC-I, with K84 being optimal. And this slide page focuses on the effect of light intensity and cell growth stage on GFP expression. The left panel D shows that the GFP expression level increased significantly with increasing light intensity under dark condition, which means that higher light intensity enhances regulation of gene expression by photoregulatory system. And the right panel demonstrates the effect of different cell growth stages, the OD600. With increasing cell density, GFP expression gradually increased under dark condition, whereas it was always lower under blue light condition. So to summarize, both light intensity and cell growth stage significantly affect the regulatory effect of the system, with GFP expression being more active under dark condition, whereas blue light inhibits the expression. This figure compares the strains under different conditions for two protein expression, and SDS pageo electrophoresis was used to detect the protein expression levels. Opto BL21 dark strain promotes protein expression under dark condition and inhibits protein expression under blue light irradiation. Comparing with that, the BL21 DE3 strain promoting expression was completely dependent on IPTG induction and was not affected by light. So we then discuss the potential application of this system. This is a precise regulation of biosynthetic metabolic pathway of 1,3-PDO by the photocontrol system. During metabolism, glycerol is first converted to 3-HPA in an enzyme catalyzed reaction with VB12. Subsequently, 3-HPA is catalytically reduced using NADPH as an electron donor to generate 1,3-PDO. The protein does not bind to LAC-O under dark condition, lifting the inhibition of T7RNAP to drive efficient expression of downstream genes. In contrast, under blue light conditions, the protein binds to LAC-O and inhibits the expression of T7RNAP, leading to the blockage of metabolic pathway. The yield was significantly higher than that of traditional strains induced by IPTG to initiate gene expression. And this is another precise regulation of biosynthetic metabolic pathway, which is EGA, using the Opto BL21 dark system. Firstly, HIS was catalyzed by EGTD enzyme to generate the HER, and HER was further converted to HER-CYS-SO via the enzyme EGTB and EGTC. Finally, the final product EGT can be generated under the catalytic action of enzyme EGTE. In the construction light control system, the protein does not bind to LAC-O under dark condition, depresses the T7RNAP, and initiates efficient expression of downstream EGT1, EGTD, and EGTE genes. In contrast, under blue light condition, binding to LAC-O inhibits gene expression, resulting in blocked EGT synthesis and decreased yield. And this is my comments on this paper. The advantages are that firstly, this research is very innovative. The second point is that the design dual system can freely choose the expression model of the photoactivation or photoinhibition according to different needs of metabolic engineering. And the third point is that the use of the photoinduction replaces the environmentally harmful and costly IPTG chemical inducer, which reduces the production cost and the risk of environmental pollution. It also avoids the chemical inducer residues. But the disadvantages are as follows. In industrialized large-scale fermentation, the light penetration capacity is limited, and the light may not be evenly distributed, which can lead to uneven gene expression. And secondly, the light control system has a certain degree of leak expression, which will affect the regulation accuracy. Meanwhile, the background expression level of a light control system in the off state was not sufficiently evaluated in this paper.